I'm going to go ahead and record this lecture in case Riley is joining this class. Okay, I have one lab safety agreement. Kai, do you have yours?
Yes.
Great, thank you. So we're going to agree to be safe in class time, right? In class lab time. Thank you. And the rest of you try to remember to bring that. Next week I'll probably send out another email.
Yeah.
With an attachment on it. I have a couple of copies if y'all want to just take a copy. It have to be signed by you and by your parents. Okay, so if you remind me in the class, I'll drop that with you. Okay. All right, so we're on 1B. So we're at the second half. Did y'all get started reading the first half, basically, of your module?
Oh.
Have you started reading? You don't have to. It's up to you. Just as long as by next time, when you come to class next week, you have read all of Module 2.
My books and everything are there when I get home, so.
They're going to be here like today or something?
Yeah.
Okay, cool. All right.
I've already read the whole module.
You've read everything.
Yeah.
Good. So just review that. And remember the study guide's at the end of the module. Questions 1 through whatever, 15, 20, whatever it is. And you can skip number 1, which is just definitions, unless your folks want you to write out definitions, which go ahead and do that. You'll turn that into me next week. You'll sit down. I'll give you, we'll change, right? I'll give you your quiz. You'll give me your study guide. You'll take the quiz, and then we'll have a lecture starting on Module 2. Okay? So that's next week. All right. So we are looking into characteristics of life here. This is one of my heroes of the scientific world. His name is Antoine van Leeuwenhoek. That's how you pronounce it, Leeuwenhoek. And Leeuwenhoek was one of the first guys to wonder what causes disease. Okay, he started wondering about disease. And he looked at a drop of water. He invented basically the first microscope. And he discovered microorganisms, living creatures that were too small to be seen with the eye, but he saw with his little scope that he had invented. So he's kind of the first microbiologist, if you will. This is what his microscope looked like. It's kind of strange looking. Notice right here in the picture, he's actually holding it in his hand. It's only about, 4 or 5 inches long. It's not very big. But it does right here, I have another picture of it right there. That's a little bit of a lens, a little small bit of glass in there that is going to be used to magnify what he's looking at. So you see right here on number 1 right here, there's a tip. And that tip, he would put a drop of water from the pond. And when he looked in there, he was amazed to see something. He called them animalcules. That was the word he used for them because he saw something swimming around in there. All right, and he said, whoa, what is this thing? That's what it looks like. So there's the little tip. He had a drip of water just hanging there, and there's that little lens right there. And he saw these he drew what he saw. And I love that because that's what biology is. We're going to draw what we see. So he saw these creatures swimming around in that water, and he thought, wow, they're living in there. Maybe, he said, maybe these are responsible for some disease. So that was kind of the first glimmer of the idea of what we know something called the germ theory, which is there are these germs that cause us to be ill. All right? So some other scientists were wanting, remember we talked about spontaneous generation? Did we talk about that last time? How, certain things come from rotting meat, for instance. Did we look at Francesco Redi's experiment last time and this kind of thing? Well, this man, John Needham, he wanted to show that spontaneous generation was indeed true. And so he devised an experiment to prove spontaneous generation to be true. Now, you know there's something wrong with that statement, right? Because does science prove anything? No, it doesn't prove anything. All right, so what he did was he took a broth, like a soup, and it was infected with some microorganisms, and then he heated it up in a flask, and then he put a cork in it, and then stuff started growing in it some more. Okay, so he said, look, I heated it, there was no growth here, and then now there's growth again. So that shows you that these microorganisms are coming from the broth. That's where that life came from that non-living broth. Okay, so some other people looked at that. They said, okay, well, I think there might be a problem with his experiment. Okay, and that problem is he may not have used what we know as aseptic technique, in other words, sterile technique. He may have introduced new bacteria into the situation afterwards. Perhaps he didn't boil it long enough. I think that's what they finally came up with, is he didn't boil it long enough. So this guy comes along. His name is Lazzaro Spallanzani. Did I say it right? Spallanzani. Okay, Spallanzani. He boiled it longer. Okay, so he took it, he boiled it longer, and then he closed the glass lid. He actually used the melting of the glass to close that lid off like that. Okay, so he says 2 days later there's nothing growing in here. But what's wrong with this experiment? What might be the complaint of Mr. Needham about this experiment? He might say, when you close it off like that, there's no air in there now. So whatever was growing needed air to breathe, so therefore you just suffocated it. All right, so that's when Spallanzani said, okay, that's good point. So this guy comes along, probably one of the greatest heroes of microbiology in the history of the world. And sad to say, there were some things he did that were wrong. But in this particular case, he was right. And he said, okay, I believe that Needham's experiment was faulty. I believe that spontaneous generation is ridiculous. Living things only come from other living things. But I've got to find a way to take that experiment, this guy's experiment, and improve it. And so here's what he did. He made the very, very famous, what's known as the swan neck flask. So notice it has a curve in it right here. Okay, the curved neck swan neck flask right here. So what his idea is, he's going to put the broth, which is growing some microorganisms, into the flask. Then he will boil it and boil it for a long period of time, and then let it sit. Now, notice the curve here. Here he is with his curve. He's going to see air can still get in there. Air can still travel into the flask, but stuff can't travel through. So microorganisms don't go through this dry, open tube right here. And so we're going to see what he ended up with. As he boiled it, he waited some time, and he found no growth in this flask right here. But in this one, he boiled it, he broke off the neck, he waited, and there was microbial growth. Now, what kind of conclusions can he draw from these two experiments right here? What happened down here? Why is there microbial growth there?
Because they can get in because there's no curvature.
Right. It fell in, if you will, right, just from the environment. You could just say, have you ever left bread out? You'll have mold growing on that bread pretty quickly. And you think, oh no, we have mold around. There's mold all around us, y'all. It's all around us. Okay, we can't get away from it. Some people get sick from it, so we have to be very careful. But you see here, he was air getting to it, no growth here. So this, once and for all time, did disprove spontaneous generation. So it was Louis Pasteur who disproved Aristotle, that many hundreds of years previous to him. It took all this stuff, all of these learning from other people's experiments to come up with this experiment to indeed show, without a doubt, that life doesn't spontaneously arise. It has to come from other life. And since that time, of course, we've seen that to be true. We can't say it's proven, but we can say that it's true. Now, is spontaneous generation theory still around today? I believe your book asked that question, and the answer is going to be yes, it actually is around. And that's because it's called abiogenesis to sound so smart. Abiogenesis. And that's the idea that long ago, millions and millions and millions of years ago, that simple life forms spontaneously appeared through chemical reactions. That's the idea of macroevolution. Okay, so it's very clear life comes from life, except in this unique time in the very, very distant past when life came out of non-living things. That's not a very scientific sentence, is it? Not very scientific. If what Louis Pasteur did was correct, then how in the world can this be true? This is spontaneous generation, okay? And science does not support this as an idea, but if you want to get around the idea that God created everything, then you got to kind of sort of buy into this. Okay, so I would say that honestly, evolution is based on something that's not scientific at all. Now, we can go test it now. We can take a theory of evolution and test it, and certain things come up that make it look pretty reasonable. But there are lots and lots of other pieces of evidence to show that it's ridiculous. I'm biased, by the way, but y'all know that already. Okay, criteria for life. This is interesting. In the old days, I used to always say there were 4 criteria for life, but we've added some now. So we're going to go with the new criteria, I guess, like this. Number 1, all life is composed of cells. Okay, so your number 1, you're going to write life. So criteria of life, number 1, all living things composed of cells. So that's the basic unit of structure and function. Some cells are very small. Some creatures, some living creatures are only one cell big. That's it. They're single-celled organisms. A yeast, for instance, is a single-celled organism. A bacterium is a single-cell organism. This little puppy over here is a multicellular organism. Lots and lots of cells. We, of course, are made up of multiple cells. Number 2, all living things grow and develop. Okay, they either increase in size or increase in number of cells, or both. So if it's a single-celled organism, it might start off very small, and that single cell gets larger, whereas this puppy's going to grow up into a dog, right? More cells and bigger. Number 3, all living things require energy. So listen to this one closely. All living things require energy to power their activities. This energy comes from the sun or from their food. The word is metabolism. So write down for this one, metabolism. And metabolism is getting energy. Getting energy. All right, so metabolism is the sum total of all chemical reactions to obtain and convert energy. So how does a plant get energy? From the sun. How do we get energy? From our Chick-fil-A at lunch today. Do you all have Chick-fil-A for lunch today? I missed the Chick-fil-A day.
I didn't have enough time to sign up for it.
Because you're not here early enough? I didn't know about it. I could have signed up for it, but I had already brought my lunch, so it'd be wasteful. Okay, so that's 3 of the criteria. Yeah, we have those 3 written down. You don't have to write every word, right? Just the notes that'll help you remember. And those cat fans out there, here we go. All living things maintain a stable internal environment by regulation. So you can just say regulations. Because some things can actually regulate their body temperature. Those things are called warm-blooded or endotherms.
Lasers.
Some things do not regulate their temperature, but they regulate other things. So there's certain types of regulation going on there. Number 5, all living things sense and respond to changes in their environment. They have sensory knowledge of the environment. And you might think, because we're talking about all living things, right? So not just, things we eat like our cat and our dog and us and this kind of thing, which makes sense. We have eyes, ears, nose, mouth, that kind of thing. But think about a mushroom. A mushroom's alive. It senses the environment. You better believe it. It knows when it's wet outside. That's for sure. It also knows which way's up. It's interesting how plants know which way is up. They know which way is down. They know where gravity is. Have you ever seen a plant that moves and follows the sun?
Sunflower.
Yeah. All plants really do it. Sunflowers are just really dramatic about it. But they respond to change in the environment, the change position of the sun. Number 6, all living things contain heredity information in their DNA. You can just write down DNA. All living things have DNA, which of course stands for deoxyribonucleic acid, which is the stuff that determines our traits, what we look like. And here it says, enables life to reproduce kind after their kind, which is out of Genesis chapter 1. Everything will reproduce its kind. So that's interesting. Is that it? Do we have 6 blanks for it?
Yeah.
Because in the old deal, one of the things was reproduction, the ability to reproduce. Which is why viruses didn't fit into this category of living things. Viruses aren't considered living things because they can't reproduce. They can only reproduce inside a host cell. And therefore they have to co-opt the machinery of the host cell in order to reproduce. So I find that interesting that that's not one of our criteria. I'm gonna have to look into that. It does have it here, enables life to reproduce. If they're putting DNA and reproduction in the same grouping, maybe. So go ahead and write DNA-reproduction. Just so we can cover that. And so I can still keep saying that viruses are not living. All right, getting energy. Here's our metabolism I was talking about right here. We have to get our energy from food. All right, so we will intake our energy through chemical carbohydrates, fats, proteins, and other things, minerals, vitamins, things like this. We will take it in. We will have our body's energy producing heat. And then, of course, we have waste that we get rid of. So the sum total of all the processes in the organism that convert energy and matter from outside sources to energy and matter to sustain life. All right, we can't get along very long without food, without our metabolism running. You have some people talk about how they have a really fast metabolism. Some people talk about they have a slow metabolism. This kind of thing. It's just really metabolism. You know, our DNA does determine how we look and this kind of thing. I'm just convinced as I get older, my metabolism is slowing down. You know, I'm kind of tired a lot. This kind of thing. Okay, so we have these two words, catabolism and anabolism. Okay, and that's just a different word right here. So we're going to see the blue arrow is catabolism. So catabolism means to use energy and chemicals and break them down. We're breaking down larger chemicals. Okay, that's to catabolize. So I eat food and I break it down with my teeth, I break it down with my saliva, I break it down with the acid in my stomach, break it all down, then I distribute it around to my body by breaking it into proteins, carbs, and fats. My proteins are broken down even further, catabolized even further into amino acids, and those amino acids can be reassembled into new proteins for my body. That's how you're going to get muscle growth. Our carbohydrates are broken down ultimately to something called glucose. Glucose is the magic of metabolism right there. Glucose is how we get energy. In my A&P class, which stands for Anatomy and Physiology, talking about human system, the answer is always ATP. ATP is energy. And it stands for adenosine triphosphate, and that's what glucose and oxygen together make this energy called ATP. I believe that's supposed to be ATP right there. And then our fats are broken down into what we call fatty acids, and those fatty acids can be used to build up and store nutrients in our body, in our bones, and in our muscle. Anabolism is what we use to create things. So the yellow arrows here. So our blue is breaking things down. Our yellow is building things up. All right, so both of these are happening in our metabolic pathways in our body. Now, photosynthesis is where all the magic begins. All right, without photosynthesis, we would not be alive. Even if you never eat any vegetables, you eat meat that eats vegetables. Okay, so if you're a, what do you call it, a cannibal? Not a cannibal, that's not the word I'm looking for. What's it called when you only eat meat?
Carnivore.
Carnivore. If you're a carnivore, you still depend on plants. Yeah, hopefully none of you are cannibals. That means you eat humans. We don't do that. So photosynthesis is the process by which green plants, all plants are green by the way, there's only one that I found that isn't green. It's called an Indian pipe brush. It's white. It's still a plant though because it has roots. Okay. Green plants, some other organisms, some bacteria, some algae will also go through photosynthesis. They take energy from the sunlight and chemicals to make food. So here we go. They take carbon dioxide and water, the energy from the sun, and they make sugar. They make glucose. And if you remember from the previous slide, glucose is what we need to make our ATP, right? So we have to have glucose, whether we get it from a protein source or get it from a carb source, we must have it. So any kind of carb is a plant. So think about grains like breads, wheat, and that kind of stuff. That's all going to be plants that's going to give you that glucose. You can get it from protein, but it's not as easy. So I wouldn't recommend being a carnivore. I would recommend being an omnivore, eating both plants and animals. And then, of course, right here, notice oxygen is being produced as a byproduct, which is very useful because we need oxygen, right? We actually take that arrow right there and point it the other direction, and that's what we're doing, the opposite of photosynthesis. Do you already know that chemical reaction right there, that chemical formula? 6 CO2 plus 6 H2O yields C6H12O6 plus 6 O2. That's carbon dioxide plus water gives me glucose plus oxygen. Is it already in your book? Is it written down for you? I would write it down, please. This is one of those things that I have students who are seniors, and they're still writing at the top of their quizzes and getting 2 points on their quiz. All right, this is so important. You must know this. You need to know what it stands for. I have 6 molecules of CO2, that's carbon dioxide, plus 6 molecules of water, H2O, which will give me 1 molecule of glucose and 6 molecules of oxygen. If I turn that around on the bottom right here, you see how I have C6H12O6, that's glucose. I had a salad for lunch today. I'm eating it right there, that's my salad. Guess what else I was doing? I was breathing. That's why we had to breathe. Okay, and I took my salad and my breath and I breathed out CO2 I also breathe that water. That's why when you go on a window, you can draw on it because water's coming out. And there's my energy for being able to stand up here and talk to y'all. My ATP. All right, we need to do that every day, right? So it's the opposite of respiration. Excuse me, the opposite is called respiration. So this is photosynthesis. This is cellular respiration. We will talk about that in a unit coming up. It's called the Krebs cycle, and it's one of those things that strikes fear into the heart of every undergraduate to have to know the Krebs cycle. So when we get to that, I'll warn you. But you don't have to know it as well as my biology 2 kids had to know it. But you will have to know it for this class. All right, but please memorize the photosynthesis equation. All right, hint, it's probably going to be on a quiz. All right, that's a big hint for you right there. And I'm guessing it's in your book as well, okay? And remember what each of those things are. All right, here we are looking at something called an autotroph. Autotroph is something that makes its own energy. And that would be plants, of course. In this particular case, we're looking at a tree. So organisms that make their own food are known as autotrophs. If an organism cannot make its own food, it's called a heterotroph. Hetero just means different. It needs to have its energy from a different source besides itself. Okay, that's where that word comes from. So our heterotrophs would be broken down into these main groups here. We have carnivores, herbivores, and omnivores. Okay, so in our carnivorous group, we have tigers, apparently a cuttlefish. Who knew? Not very cuddly. They are carnivores. Platypus, anteater, lion, shark, walrus, penguin, they eat fish, right? Herbivores are eating only plants. We have our rhino, deer, parrot, koala. So that's one. There we go. Okay. Giraffe, hippo, kangaroo, elephant. Omnivore means they eat both, some of both. So hedgehog, boars, skunks, who knew? Monkeys. I knew about bears because they eat berries and then they eat fish, right? You know. Kiwi, turtles, ostriches. Okay, so what group do we belong into? Most humans fit into the omnivorous group. Even if you choose to be a vegetarian and you're only going to be eating plants, herbivores, our teeth are designed for eating both herbs or plants and meats. Okay, more terms. Here we go. Heterotroph. Do you have these written down in your, you have blanks for them? Oh, we have our matching, right?
Yeah.
We have matching. Okay, so here we go. Heterotroph is an organism that depends on another organism for food. So what letter did that end up being?
Well, it's 9.
Oh, it's 9.
Well, it's on here, so it's 9 here, and then it's down.
Yeah, so your answer is 9 in your blank right there?
Yeah.
Okay. Our next term to know is endotherm. Endotherm is an organism that's internally warmed by heat-generating metabolic process. An endotherm is a warm-blooded animal. What number did that end up being?
It would be, so it would be, what was endotherm?
Endotherm, oh, I see what you're saying. The number is going on the very, bottom row there.
Wait, I think that is.
Organism that is internally warmed by heat-generating metabolic processes. Hi there. So you're the new student. You are Riley.
Yes.
Hey, Riley, come on in.
About it.
You want to come back here? Yeah, we start class at
2:30.
2:30. Yeah.
I forgot.
Well, it's nice to meet you. I don't. So do you have your lab book by any chance?
No.
No. Okay.
I
Some
can-
people don't have it. Yeah. So what I'm doing, Riley, is I'm recording this lecture,
Okay.
and I'll send it to you. You get me your email address,
Alright.
and I'll send it to you.
I think I already did.
Okay, I've got your folks. I don't know if you have one here for yourself or whatever. All right, the next term is called an ectotherm. That's an organism that lacks internal mechanism for regulating body heat. So that means they're cold-blooded. Ectotherm, just another word for cold-blooded. Cold-blooded it's not very accurate. It's not like they have cold blood, it's just they don't regulate their heat the way an endotherm does. And our last term is receptors. These are specialized structures that allow living things to sense conditions or their environment. Okay, so sensory receptors. For instance, our eyeball is a sensory receptor. It senses light. Our skin has sensory receptors in it. It senses touch, heat, pressure, pain, that kind of thing. All of our sensory organs, our ears, tongue, nose for smelling stuff. All right, so we got those matching done. Do we have all of them?
There's still 5 left.
Ah, well, that means I must have more. Is one of them homeostasis? Still one of them?
Yes.
All right, so homeostasis helps the body maintain a stable internal environment. Does that match up with one of them?
Yes, it does.
Okay. So homeostasis means to keep everything on an even keel. The body of any living creature is always trying to fight off stress in order to keep things normal. Okay, that's called homeostasis. We want to keep things normal. We don't want to be too hot. We don't want to be too hungry. We don't want to be too cold. You know, and every living creature has a strategy to overcome any kind of stress that may come in. So, so we have a receptor. So let's just say right now you're listening to me. You can hear my voice. So there's a receptor in your ear called the cochlea that has these special little patches of tissue in there. And it's vibrating your ear, coming to there, and it's going to your control center, which happens to be your brain. And there's a special part of your brain that can process. Not only can you hear me, you can understand the words I'm saying. They're making sense to you. You speak English, fortunately, right? And then there's an effector. Your effector says, I heard that. Now I'm going to use other nerves to send it to my hand, my muscle movement to write it down. Okay, so all that's happening from receptors. So we have a receptor, a control center, and an effector. So when you're writing like that, you have your effector right there. These right here are pictures of specialized cells called neurons. Neurons are your brain cells and your nerve cells. All right, we have asexual reproduction and sexual reproduction. In this, for asexual reproduction is when we start with one parent and end up with two offspring, or two daughter cells, they're called. All right, and they're exactly the same. Essentially what's happening in asexual reproduction is cloning. It's a cloning type of a thing. We have identical offspring. Now this is happening in you right now. Your skin cells are going through asexual reproduction, producing all the time and sloughing off when we get rid of them, right? So that's where dust comes from in your house, yes. Sexual reproduction is when you have combining DNA from two parents, and you have half from the female, that's the egg right there, and half from the male's the sperm. Those come together in order to form a zygote, which has the full amount of genetic information. Okay, so the egg and the sperm only have half of the genetic information. And this particular one shows humans. We have 23 chromosomes from the egg, 23 chromosomes from the sperm. We end up with 46 chromosomes for a full-sized cell. Okay, so most of our cells are going through asexual reproduction, and our specialized cells go through sexual reproduction. All right, so we're going to see all the different organisms we learned about in biology this year. We're going to see some operate strictly on asexual reproduction. For instance, bacteria. Bacteria have asexual reproduction. They go through a cloning process. All right, they do have ways of exchanging genetic information, however. It's very kind of fancy, specialized ways. And anytime you can exchange and share genetic information, changes can take place. And if changes take place, then we have variants, right? The word variants is out there. We have this virus that has this new aspect to it. Well, it had some genetic change, and that change happened in some way. A virus, of course, like I mentioned before, a virus is not living because it doesn't go through reproduction, but it does have nucleic acid. In the case of COVID, it has ribonucleic acid, or RNA. All right, inheritance is the process by which physical and biological characteristics are transmitted from parent to offspring. Okay, this is if somebody says, man, you look just like your dad. You look just like your mom. That's because you got those traits from your parents. We will do a genetic module. It's pretty fun. In fact, I'll send stuff home, or at least I'll have you taste something. There's this genetic thing that some people can taste and other people can't taste. And I'll literally give some to you, and you'll taste it, and you'll say, oh, it tastes terrible, or I don't taste anything. You know, so that's genetic. You know, one of them said, who's an Aggie in the room? Any Aggies? So gig 'em. Show me a gig 'em. Okay, if your thumb goes like that, it's called a hitchhiker's thumb. See how it's going way back like that? Not everybody can do that. You're doing it real good. Yeah. It's dominant. That's the dominant trait. Some people, they do this, and their thumb is just straight up and down like that. Okay, but I can do that because I have the genetic trait that's dominant that says hitchhiker's thumb. But I'm a longhorn, so hook 'em horns, man. So those traits come from your parents. So here we have a father and a mother, and look at the trait, this pink, whatever that pink is. This is a son. That's because that's called the X and a Y. Has the mother's trait, whatever it happens to be, brown eyes, blue eyes, whatever it is. A mutation is an abrupt and marked change in the DNA of an organism compared to that of its parents, something that's changed. Typically, a mutation is a negative thing. All right, it tends to confer upon the host something bad. All right, so in fact, I don't know that we could even say that a mutation has ever ended up being good. All right, unless you believe in evolution, in which case Lots of mutations had to happen in order for life to pop up out of nothing. Which I, of course, don't believe. So, all right. Are all our blanks filled in now?
Yes.
Hooray. All right, so those are all things. I covered each one of those. Now we're going to talk a little bit about the kind of things we use to study biology. By the way, Riley, my degree is in microbiology from UT, right? So I love microscopes. So we'll be looking at microscopes today. I meant to bring my big plug-in microscope, and I didn't. So we'll be sharing these microscopes today. These are battery operated, so you don't need a plug for these. All right, what are we looking at there? Y'all know what that stuff is?
Scalpels.
Mm-hmm. That's a dissection kit. And what we'll be using mostly are scalpels. I do use scissors now and then. Scissors aren't as helpful as you would think they would be, but we sometimes will use those for some macro dissections right there. Also, little probes and tweezers are very helpful. These are called T-pins. T-pins help to pin down a specimen so we can see it more easily. Namely, the one we'll be using the T-pins on will be the earthworm. We'll lay it out now, so we can see all the internal structures and things like that. Yeah, hopefully y'all aren't squeamish about dissections, but our dissections don't come until the second half of the year. We don't have any dissections until the spring. So that's different. That's how one of the ways they changed up this curriculum. Okay. We will be using metric system in science. We always do. And so therefore, you need to know some things about some common metric units. Whenever we're measuring anything, oftentimes if we're even doing a dissection, we will measure the specimen and write that down. We will be using meters, centimeters, millimeters to do that. Sometimes when we're measuring out something to pour, for instance, I have my graduated cylinder right here, and that's going to be measured in milliliters. All right, and so that's going to be, this one it says is 100 milliliters. All right, so we're measuring out 100 milliliters. By the way, I don't know if you know this, but milliliters is the same thing as cc's. Cubic centimeters, they're the same exact measurement. So if you've ever had an injection in a syringe that had cc's, like 3 cc's of something, it's the same as 3 milliliters. And I would think that this is a graduated cylinder. They're fairly easy to use. You'll pour into it and you have to pour some out and pour some back in to measure just right. I don't know why we don't just use syringes. Syringes are very accurate to measure with. All right, so that would be volume. We use volume in liters, milliliters, cubic centimeters is the same thing as a cubic centimeter, is the same thing as a milliliter. It's kind of weird to think of cubic centimeters being the same thing as something in a volume thing. Mass, we're going to be using grams, kilograms, metric ton. We'll never get anything that heavy. My scale won't measure that. And then a temperature, we're going to be using the Celsius scale, where water freezes at 0 and boils at 100. It really makes the math much easier, than trying to look at, because in the Fahrenheit scale, water freezes at 32 degrees and boils at 212 degrees. And to go from one to the other, you have to do kind of a fancy little mathematical conversion. And I just say, ask your phone. I don't know why we have to. My chemistry kids have to do the conversions, but hopefully we won't have to do that. Anyway, that's table 1.1 on page 24, common metric units. There's a possibility something of this chart will be on a quiz, and we do have a quiz next week, Riley. We already have a quiz in module 1 next week. And the book that we're using, the Apologia book, is brand new, so there's no old ones to lend out, so I apologize for that. But let me know, Apologia does have chapter 1, you too, chapter 1 online. You can read that online.
Yeah, I did get that.
You were able to see it online? Did I send it to you?
I think so.
Yeah, from me. Okay,
Oh,
good.
my mom reads
I sent it to your mom. Yeah, I think I sent it to her. Data can be reported also in tables and graphs. I'm always very ambitious for our data to go on a graph or something, but at the same time, it's really not that useful in biology. Okay? Sometimes when we're looking at certain populations or something like that, it might work, but in biology, mostly it's all about observation, right? We're looking at certain living creatures and we're observing them. And we don't really, unless we're going to maybe measure their growth, tendencies or something like this. So, but it would be nice to have our pie graphs and our bar graphs and our line graphs and all these kind of things. So whenever we get a chance, we will try to include those. All right, so we're going to be introducing ourselves to the microscope today. These are light microscopes. And each one of these is battery operated, and I checked them all and they're all working. If they end up not working, they do each also have a plug. we can plug them into the wall. The problem is, you know, where y'all are, we have to turn the tables around or something to reach the plugs and that kind of thing. So you do need to know, I have my really fancy scope that I didn't bring, that is a binocular. It's got the two things to look through. I've got actually several really nice scopes. One of them is brand new that I bought, and two other ones, people who were in the field, in research fields, gave them to me, and they're really nice. They probably need to be cleaned or something like this. They don't have a case, so I have to just carry them around. But things you need to know about it is that we'll be looking through, the eyepiece is called the ocular, and that's where, of course, you start by looking through there. These down here are objectives, and we swing those around. At least one of these has a loose objective. That one feels pretty good. And we always start on the lowest magnification and work your way up. Yeah, this one may be a little bit loose here. We have our stage. Our specimen goes on our stage right here, and this goes up and down. And you have to, of course, move the slide around in here. My fancy ones have a mechanical stage, so I wish I remembered to bring it. The diaphragm lets light in. There's a light source up to this diaphragm, which is going to let light come through right there. So here we're going to be looking for something like this. We put it under our scope, shine some light, use our objective lenses. Keep in mind, the objectives have a number on them, but this ocular also has a number on it, so you have to multiply this. I believe the oculars are all 10, and so 10 times this one's 4, that means it's 40x, so 40 times bigger than it really is. Okay, we have light microscopes like these, which we'll be using here, but there are other kinds of microscopes as well. The electron microscopes, electron scanning and transmission electron microscope. Your book's going to talk to you a little bit about these and the technology behind them, and it's pretty fancy stuff. They're kind of, you can see here, it's like a whole desk worth. They're big. We don't have them. I never have used one. UT has, and University of Texas has a really nice electron scanning microscope. And I was a microbiology major, y'all, and I never got to use the electron scanning microscope. They were just too expensive. They didn't want students touching them. They showed me data from it, but I never got to actually go look at it. And then I don't know if this is newer, this transmission electron microscope, I've never even paid attention to those. Okay, we're gonna watch a little video. It goes on for a little bit, but just pay attention.
We just learned all about light microscopy, which was the standard until the middle of the 20th century and remains tremendously useful today.
Absolutely.
But as we began to learn more about the microscopic world, using light microscopes that could visualize individual cells, we started to become curious about structures that are smaller than a cell, some details of which were difficult to visualize and study under standard light microscopes. Fortunately, electron microscopy, developed in the 1930s and 40s, proved to be much more powerful and allowed scientists to view these smaller structures. Some electron microscopy techniques employ a resolution of 0.2 nanometers, which is more than 1,000 times better than light microscopy. With this higher resolution, electron microscopes are able to magnify their specimen up to 1 million times their original size, allowing researchers to study proteins inside of cells and the molecular structure of viruses, all of which led to a better understanding of the microscopic world. Of course, we have to ask, how do they do this? Well, the answer is in the name. They use beams of electrons, which interact with the sample as the beam is transmitted through the specimen. As we recall from our study of chemistry, electrons are stable subatomic particles that carry a negative charge. Electron beams have a much shorter wavelength than the visible light that light microscopes use, which we refer to as the de Broglie wavelength, as we learned in the Modern Physics series. This shorter wavelength allows for a higher level of detail and resolution in imaging. One type of electron microscopy is called transmission electron microscopy, or TEM. For TEM, specimens are fixed on a support grid and either embedded in resin or stained with a material like uranyl acetate or compounds of heavy metals like osmium tetroxide. Certain structures within the specimen will take up more of the stain than other parts of the specimen, and those sections are called electron-dense regions. When electrons pass through electron-dense regions, they lose more energy compared to when they pass through sparser regions. This phenomenon is used by TEM to visualize the specimen at high levels of resolution and magnification. For the stained specimen to be viewed by TEM, it must be dehydrated, embedded in plastic resin, and cut into ultra-thin slices with a glass or diamond knife. The sections must be ultra-thin so that the electrons can pass through them. When the specimen is secured in the specimen holder, an electron beam is generated and shot out of the electron gun. The electron beam is accelerated down the column of the microscope after passing through the anode. The electron beam then passes through the specimen, once again losing energy as electrons pass through electron-dense regions. The electrons then pass through the objective lens, magnifying the image, before the electrons pass through the projector, or ocular lens, which magnifies the image further and projects it onto the fluorescent screen. This is where we start translating the electrons into an image. The fluorescent screen is coated with a chemical which appears as bright spots when electrons come into contact with it. The intensity of the bright spot depends on how much energy the electrons have when they hit the fluorescent screen. The camera under the fluorescent screen then captures the image made from the bright spots, which is displayed on a computer screen. Transmission electron microscopes are used to visualize structures that are too small to study with light microscopes, such as the cell organelles, proteins, cytoskeletal filaments, and the components of cell membranes. But even with the unmatched resolution and magnification abilities of TEM, it still does have its limitations. Some of these include the fact that you cannot study living specimens or image 3D structures, because the specimens must be dehydrated and cut into ultra-thin slices before imaging. The second type of electron microscopy is scanning electron microscopy, or SEM. With SEM, specimens are coated with vaporized gold or palladium ions, because when electrons come into contact with this coating, they cause atoms on the surface of the specimen to emit electrons. These emitted electrons are called secondary electrons, and these are what SEMs use to visualize their specimen. After the coated specimen is secured on the stage of the SEM, the process starts out similarly to TEM in that an electron beam is shot out and accelerated towards the specimen. In the SEM, the electron beam then sweeps or scans the surface of the specimen, which causes it to discharge secondary electrons. There are two types of detectors in the sample chamber: the secondary electron detector and the backscatter electron detector. The backscatter electron detector picks up backscatter electrons that come from deeper regions of the sample that are a result of elastic interactions between the beam and the sample. The results from this detector are primarily used to look at crystallography and the magnetic field of the sample, but for the purposes of microbiology, researchers focus on the detection of secondary electrons, which allow them to visualize the topography of the sample, or the distribution of parts and features on the surface of the sample. The detectors convert the electron signal into a light signal that is amplified by passing through a photomultiplier before the final image is projected on a computer screen. SEM imaging is primarily used to study the topography of cells and other similar structures. With this type of microscopy, researchers are able to investigate the depth of structures, which isn't possible with TEM, as SEM produces a 3D image opposed to the flat image from TEM. Yet, just like TEM, SEM cannot visualize live specimens, and SEM has worse resolution than TEM. Both TEM and SEM are expensive and require a lot of training, so they are not likely to be used apart from those pursuing a microbiology career. We covered quite a lot in this tutorial, so let's briefly summarize. Electron microscopy has around 1,000 times better resolution than light microscopy, because electron microscopy uses electron beams instead of light, which have a shorter wavelength. This allows some electron microscopes to visualize specimens at 1 million times their original size. Transmission electron microscopy, or TEM, records the differing energies that electrons possess after they pass through a specimen stained with heavy metals to visualize small, detailed structures, like proteins inside of cells, and molecules associated with viral particles. Scanning electron microscopy, or SEM, captures the secondary electrons that are emitted from the surface of samples coated in vaporized gold or palladium ions to visualize the topography of the specimen. And the main drawback, as we mentioned, is that both SEM and TEM cannot be used to visualize living specimens, and they are also quite expensive and complicated techniques to use. And with that, we know a bit about electron microscopy.
All right.
Thanks for watching, guys. Subscribe to my channel for more tutorials.
And the scanning electron microscopy, the SEM, is very much the one that is used more commonly. Okay, so here it is. This is summing it up. I don't know what kind of notes you were supposed to write. What kind of notes were you supposed to write? So SEM, scanning electron microscopy, basically we're looking at the surface, and then we can look at internal structures on TEM, but only by getting thin samples. Okay, so we can't bore into it like with having a whole specimen and looking into it. We have to get a slice of it. So the main differences here would be that we're looking at that really cool surface specimen, but I will say, even though the SEM is not as specific or as the TEM, the SEM is real. Some of the best pictures I'll be showing you all year will be from scanning electron micrographs that were taken, and of course it's a computer that we get to see it from. Okay, so it's time for lab. We need to know a few things. All right, you're going to follow instructions. We're gonna actually look at some stuff under the scope. You're going to end up drawing that. We're going to make a slide that you're going to look at under the scope as well. All right, so we're going to learn the parts of the microscope. I'm unfortunately having to work on this one a little bit. It is giving me some trouble. Some little, one of the little lenses in there is crooked, and so you can't see a full thing on it. So it looks like I'll only have the 3 to work with. We will have to share. So we know our parts here. We'll be using a monocular one, so just a single eye here. I will tell you that students, when I bring the binocular ones, tend to not look through both anyway. They usually close one of their eyes because it's kind of hard to get it to be just right in your eyes right there. But I'll bring that eventually for you all to see, especially when we talk about bacteria. We need to look at bacteria with a very strong objective, 1000x, and it's called something called oil immersion. And I have that I'll bring which is not until the spring, actually, that we study our bacteria. We'll be using microscope slides. Microscope slides, as you know, are made of glass. All right, so if you drop them, they'll break. They will cut you, so be careful with those. They're fairly thick, I guess, but they do break rather easily. Also, you can break your slide by using the scope improperly. If you bring the objective down and it touches the slide really hard, it will break the slide. And in our college class, if you ended up breaking a slide, that was like one of the worst things you could do. It's like, it was super embarrassing. You know, it was like, oh no, you broke a slide, you know, amateur, you know, this kind of thing. So, so be careful not to do that. These right here are prepared slides. So we'll be looking at prepared slides. These are slides that somebody else made that I purchased. I will tell you, these are really, strangely, very expensive. Okay, so don't break my prepared slides. They're quite expensive. There are some that you can buy one single slide, $12 for one slide, depending on the specimen that's on there. And you might think, well, $12. Yeah, but if I'm buying one for each, you know what I'm saying, that adds up, right? So they are actually quite expensive. We are going to make a wet mount today. So I'm going to teach y'all today how to make a wet mount, where you're going to take a plain microscope slide. You're going to put a little drop of water on there. You're going to pluck a little hair out of your head and put it on the slide, and put a cover slip on the top of it, and you're going to take a look at your hair under the microscope. Okay? So that's going to be our wet mount today. I don't think we're doing a dry mount today, which is because I just don't think I think we're running out of time. We will eventually do this, where you're going to take a sample. Here we're talking about microbes, you know, so we're looking at bacteria, which we will have bacteria in this class eventually. We'll put it on a slide, you let it dry, you cover it with a certain kind of a stain, and then you let it dry again and you look at it. And so in that particular case, like when we look at your hair, it won't be stained because your hair already has some color, and we'll be able to see that. Although you might be surprised how little color it has when you shine light through it. But with these kind of things, we usually, to see cells better, we'll put a stain on them. Hanging drop slide, we won't use that today either, but we have a specialized, these are also very expensive. They have a little indention in them. and you're gonna put a drop of liquid right there, similar to Von Leeuwenhoek's little microscope that he made. We'll have a drop suspended, and we'll look at it under the scope, and you'll see things swimming around. So that'll be pond water. Does anybody have a pond? Okay, when it comes time, I might ask you to bring some pond water with you in a jar.
I have
Okay.
water behind my house.
Yeah, you do have a pond? Yeah,
Pretty
so,
much.
so I might be asking y'all to bring some pond water when it comes time for that. And we're looking at the protozoans. All right, so make sure you understand what the magnifications are. Remember, if I have 4x, I also have 10 on the objective, so 4 times 10 is 40. That means it's gonna be 40 times bigger than it really is. That's our low power. At least the 3 slides I have under the 3 scopes that are looking good right now, those are all best viewed under 40x because the specimen's pretty large. Sometimes you'll be looking at something very small and you want to bring it up to the next magnification, which would be 100x or 100 times. That's the 10x times 10. And then our what this is known as high dry power, is when you bring it up to 400. We probably won't use that today only because you won't see anything. You'll just see some color or whatever, and that's it. And then there's of course 1000x, but that has to be used with a special kind of a thing called oil immersion. You put a drop of oil in there, and you bring the oil works as another little bit of a lens, and so that's why high dry versus oil immersion. All right, let's get started. So the first thing you're going to do now, I think your book tells you you're going to look at the letter E or whatever. I don't have that, so we're just going to look at whatever I give you, and you're going to tell me what magnification it is, and then you're going to draw it in your lab book. I'll be working on this scope right here to make sure, see if I get it working. All right, so Riley, if you'll just sit closer here to Preston, y'all can share that scope. Guys, y'all can share this scope. And Kai, looks like you get your own scope, huh? So first thing you're going to do is read what is on the slide. What does the slide say you're looking at? It's written on the slide, so take a look at it. Okay, so y'all, but 2 of you have the crab larva. It's the crab larva. And so, yeah, take a look at that. Probably the best view of that is going to be on 40x. And then, Kai, do you have a paramecium or a what is it?
Planaria.
Planaria, okay, which is a flatworm, also fairly large. If you can see it when you just look at the slide itself, you know it's pretty large. I'm trying to see if I can find something to stick down in here.
Is it already set?
So you need to use these large, this is called
Yeah.
the coarse focus. Use those to bring it into focus. I think I put a couple of them into focus, but
Let me see your eyelashes.
what is going on with this thing?
Yeah, that's focused. Here about, I think it's red.
So we put what we see on here?
Yes, so your first row, does it say letter E?
Yeah.
So just skip that one. What does the next one say?
A thread slide?
Okay, that's gonna be your hair. So what's the bottom one say?
Prepared slide.
That's what you're looking at. Okay, so you're gonna tell me what it is,
Yeah.
okay? And does it say to look at it under 40?
Hey, y'all do the size thing, maybe go lower, cause it made it more clear.
Yeah, I know, we did.
Did you change the objective?
Yeah.
Like this?
Yeah.
I might not have had it on the right objective
So,
for you, yeah.
magnification.
And yeah, see which one looks the best. And you'll have to move it around with your hand, sadly, but
So,
instead of a mechanical, this really bugs me.
so you put here what we're looking at?
Yeah,
Different-
and draw what you see, and tell me at 40x, the crab larva looks like this.
It's head or
My eyelashes.
I think it's like it's in the.
I think it's right before the tail, like the tail's too long.
Try to draw that.
Or is it do the other
And then if you do go up to the next level, maybe key in on a spot that is, thinner. you know, maybe some of the little tentacles of the critter or whatever.
Something like that, because so you, yeah. Something like that. Not exactly what it looks like, but it's similar.
Good enough.
Yeah. I'm not good at details.
I think that's the most visible thing. I am not good. I am not good.
In the bottom right,
I am not
that's a
good.
big black hole.
Okay, so y'all are drawing that on the lowest row of your book.
Oh, I forgot the arm thing.
Dave, where's your book? Your lab book?
I don't know that.
It just hasn't come yet?
I only see 3.
I'm sure you can read it.
Yeah, I don't, so,
Alright, and once you're finished with that, instead of the letter E, I'm gonna give you
so it's
something
in
else.
focus, but I don't know,
So Heidi, would you prefer a sunflower?
is that the thing that
Or
moved it?
I don't-
honeybee leg? Or onion?
Oh yeah, that's focus.
So you have sunflower or onion? Any preference?
That is that.
Sunflower,
It's like
is it
focus, I
sunflower
can kinda see what it
stems
is.
or onion stem? And this is gonna go in that top row instead of the letter E.
So do we put that one on next?
Yeah, so did you try to go up a level just to see if there's anything else you could see on that?
I did not go up on a level.
Yeah, try that, go up to the next level.
Do I need to move this down?
Just do that, and then,
Yellow looks
Okay.
yeah. So you can see by the length of it, yeah, the longer it is, the more,
And then we move this
yeah.
to
Yes,
focus, or
and
we
so,
move-
so notice if you move this one, it's really big movements, but this is really fine.
Oh,
movements,
okay.
Okay, so you can do it that way. And I'm bringing you a blank slide, so this is the next thing you're gonna listen to, you're gonna look at, it's gonna be under this thing called letter E. And then this is gonna be for your hair, your wet mount that you're gonna make. So I worked in a clinic, you know, a veterinary clinic for many years, to pay my way through college, and,
It's kind of focused.
I had to make wet mounts and dry mounts all the time as part of the laboratory procedure there. So that's a clinical stuff you're learning about here.
Yeah, no, this is, oh wait, no, no. Yeah, front of that.
Yeah.
Sorry. Mixed up with it.
It's so weird because it's like different depths, it looks like.
Yeah, so it should be much closer, right? So
Yeah.
you're keying in on like that face or whatever, the head or.
I'm not on the head, I don't think. It
Okay,
doesn't
the
look
tail
like it.
end?
Yeah.
Something that shows some definition.
Work for the body.
It's kind of like aimed right here. There's like 4 on the back. It's kind of
Also, if the light seems like it's too bright coming in, you can change that. You can change the level of the light, and I believe on this one, you actually have a dimmer switch right along here.
Oh, that's the
And
dimmer.
so, yeah, if it's too bright, if it's like hurting your eye or whatever, you can bring that down. Sometimes, yeah, that'll change for yours, that's actually has holes in it that allows different amounts of light to come in. Alright, so if you wanna just change that around and see which one gives you better definition, you know, I tend to like lots of light coming through. The more light, the better, kind of thing.
Yeah, I think it's like the back area, like the tail.
Aren't there
Well,
other
there's
job only the left side?
Are we supposed to use the sunflower on there?
Yeah, that's gonna go in this top one for the letter E.
Okay.
Yeah.
Should we do that now, or should we?
Sure, go ahead, and then then I'll talk about,
Yeah,
All
so
the
I
way
have
to
to-
see them.
So how do you do it?
so now just those little clips, yeah, pop it up like that,
So I just
move to the side, and just pull it out. And that way you can put those, push those clips up like this.
Can we move this up to 4 real quick?
Yeah, it's always good to start on the lowest, is that the lowest one?
Yeah.
Right. There you go.
Does that go up in the letter E?
Yeah, it's gonna go up, yeah, just scratch out the letter E and say that is, what did I give you? The onion-
Sunflower.
Sunflower stem, I think. I will tell you that plant stems are really cool looking. Plant roots are super cool looking. And of course, keep in mind it's been stained, so was the critter you looked at was also stained, so it wasn't naturally that pinkish color.
This thing just looks like a brick wall.
Okay, so that one you might wanna go in, you know, into a higher magnification to see. You should be able to see,
Yeah.
you said brick wall, so it's squares, right? And is there a dot in the middle of it?
Is there a dot in the middle of it?
Like it's just.
Should be.
I
The
think I can
nucleus should be
see it.
visible.
Does not look like a brick wall.
Actually, maybe. It looks like, you see anything where it's like a crystal, like it's
There's
like
like, it looks like a
counter
brick
balance,
wall,
yeah.
and there's a bunch of dots in the middle of the bricks.
Okay, so the little dots in there are the nucleus. The nucleus is holding the
Do you
DNA
see it?
Yeah, I can
of
see
the
it
onion.
now.
That, by the way, is how they came up with the word cell, because it looks like cells that a monk would live in. Oops, this is still going.